We have been searching for alien life in distant star systems for decades, scanning radio frequencies, analysing exoplanets thousands of light years away, building telescopes to peer into the depths of space. Meanwhile, we might be completely overlooking something extraordinary. Alien life could be hiding right here, in our own solar system, not on distant worlds circling foreign suns, but on moons and planets we have already visited. This is not science fiction. This is a serious scientific hypothesis, gaining support from researchers at NASA, the European Space Agency, and universities worldwide. The evidence is mounting, and it is changing how we think about life in the universe. In 2005, the Cassini spacecraft detected something unexpected near Saturn: plumes of water vapor erupting from the surface of Enceladus, one of Saturn's moons. These were not small emissions. They were massive geysers shooting hundreds of kilometers into space. Analysis revealed the plumes contained salt water, organic molecules, and hydrogen, all the ingredients necessary for life as we know it. Scientists were stunned. Enceladus is tiny, only 500 kilometers in diameter. Its surface is frozen solid at minus 200 degrees Celsius. But beneath that ice shell lies a global ocean, liquid water kept warm by tidal forces from Saturn's gravity. The same gravitational squeezing that creates the geysers. This ocean is not small. It might be 10 kilometers deep. That is deeper than Earth's deepest ocean trenches. And it has been liquid for potentially billions of years. Long enough for life to emerge. Long enough for evolution to create complex organisms. Long enough for an entire alien ecosystem to develop in complete darkness beneath the ice. Then in 2015, NASA's New Horizons spacecraft flew past Pluto. Scientists expected a dead, frozen world. What they found shocked everyone. Pluto is geologically active. Its surface shows signs of recent ice flows, mountains made of water ice, as hard as rock in those temperatures. And most intriguing, evidence suggesting a subsurface ocean might exist even at Pluto's distance from the Sun. If Pluto, 5 billion kilometers from the Sun, can have liquid water, then our assumptions about where life can exist need revision. Europa, Jupiter's moon, has fascinated scientists since the Voyager missions in the 1970s. Its surface is covered in ice crisscrossed with cracks and ridges, but beneath that ice lies an ocean larger than all of Earth's oceans combined. We know this from gravitational measurements, from studying how Jupiter's magnetic field interacts with Europa, from analysing the way the ice shell flexes and cracks. The ocean might be 100 kilometers deep. It touches a rocky seafloor where hydrothermal vents could provide energy and nutrients. On Earth, hydrothermal vents support thriving ecosystems in complete darkness. Giant tube worms, blind shrimp, bacteria that get energy from chemicals rather than sunlight. If life emerged at Earth's vents, it could emerge at Europa's vents. In 2019, researchers analysing old Galileo spacecraft data made a startling discovery. They found evidence of water vapour plumes erupting from Europa's surface, just like on Enceladus. This means we might be able to sample Europa's ocean without drilling through kilometres of ice. A spacecraft could fly through the plumes, collect samples, analyse them for signs of life. NASA is planning exactly this mission. The Europa Clipper will launch in the 2024 and arrive at Jupiter in 2030. It will make dozens of close flybys, studying the ice, mapping the surface, searching for the best places to look for life. Mars was once a very different world. We know this from decades of exploration, from rovers examining rocks, from orbiters mapping ancient riverbeds. 4 billion years ago, Mars had liquid water on its surface. Lakes, rivers, possibly even oceans. The atmosphere was thicker, the temperature was warmer, conditions were suitable for life. Then something changed. Mars lost its magnetic field. Solar wind stripped away the atmosphere. The water evaporated or froze. The surface became the cold, dry desert we see today. But life, if it emerged during Mars's wet period, might not have died. It might have gone underground. On Earth, life exists kilometers below the surface. In mines, in deep rock formations, in aquifers isolated from the surface for millions of years. These organisms do not need sunlight. They get energy from chemical reactions in the rocks. If Martian life went underground when the surface became uninhabitable, it could still be there, waiting in subsurface aquifers, in pockets of liquid water, kept from freezing by geothermal heat. NASA's Perseverance rover is currently exploring Jezero Crater, the site of an ancient lake. It is collecting samples that will eventually be returned to Earth. Those samples might contain fossilized microbes, direct evidence that life once existed on Mars. But finding current life would be even more significant. The rover cannot drill deep enough to reach subsurface water. Future missions will need to go deeper, to drill into the ice at the poles, to explore caves and lava tubes that might harbour protected environments. Titan, Saturn's largest moon, is the only moon in our solar system with a thick atmosphere. It is also the only place besides Earth with stable liquid on its surface. But Titan's lakes and rivers are not made of water. They are liquid methane and ethane, hydrocarbons that are gases on Earth but liquids in Titan's frigid temperatures. This seems like an impossible environment for life, but scientists are not so sure anymore. In 2015, researchers proposed that life on Titan might use completely different chemistry than Earth life. Instead of water as a solvent, Titan life might use liquid methane. Instead of oxygen for metabolism, it might use hydrogen. The chemistry would be utterly alien, but it might work. Cornell University scientists even designed hypothetical cell membranes that could function in liquid methane. They called these theoretical cells azotosomes. Mathematical models suggest they could be stable, could grow, could reproduce. NASA is planning the Dragonfly mission to Titan, a nuclear-powered rotorcraft that will fly through Titan's atmosphere, landing at multiple sites, analysing the chemistry, searching for signs of life or prebiotic chemistry. Launch is scheduled for 2027. Arrival in 2034. Venus is the last place you would expect to find life. Its surface temperature is 465 degrees Celsius, hot enough to melt lead. The atmospheric pressure is 90 times Earth's. The air is sulfuric acid. Nothing we know of could survive there. But in 2020, scientists detected phosphine in Venus's atmosphere. Phosphine is a gas that on Earth is only produced by life or by industrial processes. There is no known natural process that could produce phosphine in Venus's atmosphere. The detection was controversial. Other scientists questioned the data, proposed alternative explanations. The debate continues, but it raised an intriguing possibility. Maybe life on Venus does not live on the surface. Maybe it lives in the clouds. At an altitude of 50 kilometers above Venus, conditions are remarkably Earth-like. The temperature is around 30 degrees Celsius. The pressure is similar to Earth's surface. The clouds contain water droplets. If microbes could survive in those clouds floating on air currents, reproducing in the water droplets, they might form an aerial ecosystem. We know this is possible because microbes on Earth live in clouds. They have been found at high altitudes, reproducing, surviving in water droplets. If it happens on Earth, it could happen on Venus. The implications of finding life anywhere in our solar system would be profound. It would mean life is not rare, not a cosmic accident that happened once on Earth, but a common phenomenon, something that emerges wherever conditions allow. If life arose independently on Earth and Mars, or Earth and Europa, or Earth and Enceladus, then the universe must be full of life. Billions of worlds with organisms we cannot imagine. The question is no longer whether alien life exists. The question is, how close is it? And what happens when we find it? The search is no longer focused on distant stars. It is focused on worlds we can reach with current technology, worlds we will explore in the next few decades. The answer might be closer than we ever imagined. Europa does not look like a place where anything could live. From space, it appears as a smooth ball of cracked ice. Its surface temperature never rises above -160 degrees Celsius. Radiation from Jupiter bombards it constantly. Any organism exposed on the surface would be sterilized within hours. Yet beneath that hostile exterior lies one of the most promising environments for life in the entire solar system. The evidence comes from multiple sources. Gravity measurements show Europa's interior is layered. A metallic core, a rocky mantle, then a layer of water estimated at 100 kilometers thick. The ice shell on top is relatively thin, maybe 15 to 25 kilometers. Beneath it is more water than exists on Earth. All of it liquid, all of it kept from freezing by tidal heating. Jupiter's immense gravity squeezes Europa as it orbits. Other moons pull on it too. Io, Ganymede, Callisto. These gravitational forces flex Europa's interior. That flexing generates heat. the same way bending a metal wire back and forth makes it hot. This heat is enough to keep the ocean liquid, despite Europa being 5 times farther from the Sun than Earth. The ice shell itself tells a story. Its surface is covered in features that reveal the ocean beneath. Chaos terrain, where the ice has broken apart and refrozen in jumbled blocks. Long ridges running for hundreds of kilometers. Cracks that show the ice is constantly breaking and reforming. Some areas look like ice flows on Earth's Arctic Ocean. Frozen, then thawed, then frozen again. This happens because the ocean beneath is not static. It has currents, convection, movement that stresses the ice above. The most exciting discovery came from the Hubble Space Telescope in 2012 and again in 2016. Hubble detected what appeared to be water vapor plumes erupting from Europa's surface, reaching 200 kilometers into space. These were not constant. They appeared intermittently, possibly linked to Europa's position in its orbit. When tidal forces are strongest, cracks in the ice open. Water from below escapes, freezes instantly, falls back as snow. This is extraordinarily important for the search for life. It means we do not need to land on Europa and drill through 20 kilometers of ice to sample the ocean. We can fly through the plumes, collect particles, analyze them with onboard instruments. If the ocean contains life, the plumes might carry microbes or organic molecules we can detect. What would Europa's ocean be like? Scientists have built models based on what we know. The water is salty, probably similar to Earth's oceans. The chemistry is complex. Magnesium sulfate, sodium chloride. Other dissolved minerals from the rocky seafloor. The ocean floor itself is where things get interesting. On Earth, some of the most productive ecosystems exist in complete darkness at the bottom of the ocean. Hydrothermal vents, cracks in the seafloor where heated water rich in minerals pours into the cold ocean. These vents support communities of organisms that do not need sunlight. They get energy from chemicals, hydrogen sulfide, methane. The bacteria use these chemicals the way plants use sunlight, through chemosynthesis instead of photosynthesis. Everything else eats the bacteria, or eats things that eat the bacteria. Giant tube worms cluster around the vents. Blind shrimp, strange fish, octopuses, an entire ecosystem powered by chemistry rather than solar energy. Europa almost certainly has hydrothermal vents. The tidal heating that keeps the ocean liquid also heats the rocky core. Water circulates through cracks in the rock, gets heated, dissolves minerals, rises back to the ocean floor carrying nutrients and energy. If this is happening, Europa's ocean floor could be covered in vent systems. And if vent systems exist, life might exist around them. But would Europa life be anything like Earth life? That depends on whether Europa life arose independently or whether it shares a common origin with Earth life. This might sound impossible. Europa and Earth are separated by 800 million kilometers, but exchange of material between planets is possible. We know this because we have found meteorites on Earth that came from Mars. When a large asteroid hits a planet, it can eject material into space. Some of that material eventually falls on other worlds. This process is called panspermia. If a rock containing microbes was ejected from Earth billions of years ago, travelled through space and landed on Europa, those microbes might have survived. Bacteria can survive in space for years, possibly much longer if protected inside a rock. They can survive the heat of atmospheric entry. They can survive impact. If Earth microbes reached Europa and found themselves in a liquid ocean, they might have established themselves, evolved, diversified. Europa life could be distant cousins of Earth life, using the same DNA, the same basic biochemistry, just adapted to a very different environment. Or Europa life might have arisen completely independently. In that case, it could be utterly alien. Different biochemistry, different genetic system, maybe not even using DNA at all. This would be even more scientifically valuable. It would prove life can emerge through multiple pathways. That Earth solution is not the only solution. The Europa Clipper mission will not answer these questions definitively. It cannot detect life directly from orbit. But it can map the ice shell in detail, identify where the ice is thinnest, locate the most active plumes, measure the ocean's depth and composition, find the best targets for a future lander mission. That lander mission is already being planned. It would touch down near a plume source, deploy instruments to analyze the ice, maybe even drill through the surface layer to access fresher ice from below. The ultimate mission would be a submarine, a probe that melts through the ice shell and explores the ocean directly. This is technically challenging but not impossible. Designs exist for nuclear-powered melting probes, for submarines that could navigate in complete darkness using sonar, for systems that could survive the pressure and collect samples. Such a mission would not launch before the 2040s, but it is being seriously considered. The scientific payoff would be immense. Direct exploration of an alien ocean, direct search for alien life. Some scientists worry about contamination, Earth microbes hitching a ride on a spacecraft and introducing life to Europa. This would compromise the search for native life, make it impossible to know if anything we found was actually from Europa or accidentally brought from Earth. NASA takes this seriously. Any mission to Europa must be sterilized to extraordinary standards. Heat treatment, radiation, chemical cleaning, reducing the number of viable microbes to acceptable levels. The Europa Clipper will not land. It will not even touch the plumes, just fly through them and collect particles. This minimizes contamination risk, but future missions that land or penetrate the ice will need even stricter protocols. Planetary protection is not just about protecting Europa. It is about protecting Earth too. If Europa has life, that life might be dangerous to Earth life. Probably not. Almost certainly not. But we cannot know until we study it. Any sample return mission would need to treat Europa material as potentially hazardous. Containment, quarantine, analysis in sealed facilities. These precautions would be expensive and time-consuming, but necessary. The discovery of life on Europa would change everything. It would tell us life is common in the universe, that oceans beneath ice are habitable, that perhaps dozens or hundreds of worlds in our galaxy harbour life in subsurface oceans. It would raise questions about consciousness, intelligence, whether evolution in Europa's ocean could produce complex organisms, maybe even something we would recognise as intelligence. We will know soon. The Clipper launches in 2024, arrives in 2030, begins its flybys, collects data, sends it back across the solar system. Within a decade, we might have our answer, not from a distant star, from a moon of Jupiter. A world we have known about for centuries, but are only now learning to truly see. Titan is unlike anywhere else in our solar system. It is the only moon with a thick atmosphere, the only place besides Earth with stable liquid on its surface, the only world where rain falls, where rivers flow, where waves lap against shorelines. But everything about Titan is alien. The atmosphere is nitrogen and methane. The temperature is -179 degrees Celsius. The liquid in the lakes and seas is not water. It is liquid methane and ethane, hydrocarbons that on Earth are gases we burn for fuel. On Titan, they are oceans. The Cassini spacecraft spent 13 years studying Titan. What it revealed was astonishing. Titan has hundreds of lakes, 3 major seas larger than North America's Great Lakes, river systems that drain highlands and flow toward the seas, coastlines with bays and inlets, islands, even waves detected by radar. The landscape looks eerily Earth-like when you see it in images, until you remember that the rocks are water ice as hard as granite in those temperatures. The lakes are methane. The rain that falls is methane. The whole hydrological cycle is based on methane instead of water. This seems like an impossible environment for life. Water is essential for life as we know it. It is the universal solvent, the medium where biochemistry happens. Every organism on Earth is mostly water. How could anything live in an environment with no liquid water at all? Scientists started asking a different question. Could life use methane the way Earth life uses water? In 2015, researchers at Cornell University explored this possibility. They designed hypothetical cell membranes that could remain stable in liquid methane. On Earth, cell membranes are made of phospholipids. These are molecules with a water-loving head and water-hating tail. They arrange themselves into double layers, with the heads facing outward toward water and the tails facing inward away from water. This creates the boundary of the cell, but phospholipids would not work in liquid methane. They would dissolve or fall apart. The Cornell team proposed molecules made of carbon, hydrogen, and nitrogen that would be stable in methane. They called the resulting cell structure. Computer models suggested azotosomes could work. They could form stable membranes, contain chemical reactions, grow, divide, reproduce, everything a cell needs to do. The metabolism would be completely different from Earth life. On Earth, organisms ultimately get energy from chemical reactions involving oxygen. We breathe oxygen, combine it with food, release energy. Even organisms that do not use oxygen directly still evolved in an oxygen-rich environment and depend on oxygen-based chemistry. Titan has almost no oxygen, but it has hydrogen. Titan life might breathe hydrogen instead of oxygen. It would inhale hydrogen from the atmosphere, combine it with carbon compounds, release methane as waste. This would be the reverse of Earth's oxygen-based metabolism. The chemistry is possible. Whether it actually happens is unknown. Titan's chemistry is incredibly complex, even without life. Methane in the upper atmosphere is broken apart by ultraviolet light from the sun. The fragments recombine into larger molecules. Ethane, propane, complex hydrocarbons with dozens of carbon atoms. These molecules rain down onto the surface, accumulate in the lakes, settle on the ground. Over billions of years, Titan has been making organic molecules. The ground is covered in them. The lakes contain them. If life exists on Titan, it has endless raw material to work with. The Cassini mission found something intriguing. Hydrogen disappears near Titan's surface. Models predict hydrogen should be abundant in the lower atmosphere. It is produced by chemical reactions in the upper atmosphere and should sink. But measurements show less hydrogen near the surface than expected. Something is consuming it. One explanation is life. Organisms breathing hydrogen, using it for metabolism, converting it to methane, which then evaporates back into the atmosphere. This is not proof. There are other possible explanations. Surface chemistry, underground absorption, unknown atmospheric processes. But it is consistent with life. Another mystery is the lakes themselves. Some of them show unusual features, edges that change, bright spots that appear and disappear, smooth surfaces that should be rougher. These could be geological processes, or they could be biological, films of organisms on the surface, blooms of methane-based plankton. We do not know. Dragonfly will be our next mission to Titan. A rotorcraft the size of a car, powered by nuclear battery, capable of flying hundreds of kilometers across Titan's surface. It will launch in 2027, arrive in 2034, spend years exploring. The mission is designed to study Titan's chemistry, to understand how organic molecules form and evolve, to search for signs of prebiotic chemistry or life. Dragonfly will land at multiple sites, scoop up samples, analyze them with onboard instruments, look for complex organic molecules, search for patterns that suggest biological processes. It will visit Selk crater, where an asteroid impact melted the ice and mixed it with organic compounds, creating a temporary environment where water and organics coexisted. If life can start on Titan, that might be where it started. One of the biggest questions is whether Titan has liquid water. On the surface, no. Water is frozen solid. But beneath the surface, like Europa and Enceladus, Titan might have a subsurface ocean. Gravity measurements suggest this is possible. If Titan has both a subsurface water ocean and surface methane lakes, it has two separate environments where life might exist: water-based life underground, methane-based life on the surface. This would make Titan the only known world with two independent biospheres. The implications for finding life on Titan are profound. If life can use methane instead of water, then the number of habitable worlds in the universe expands enormously. Every cold moon or planet with hydrocarbons becomes potentially habitable. Pluto might have life, Triton around Neptune, countless objects in the outer solar system, the moons of exoplanets orbiting distant stars. The universe could be full of methane-based ecosystems we never considered possible. But there are skeptics. Some scientists argue that methane-based life is too speculative, that chemical reactions in liquid methane are too slow, that metabolism would be too inefficient, that evolution would take impossibly long. They might be right. We will not know until we look. And Dragonfly will look. It will answer questions we have been asking since Cassini first revealed Titan's lakes. It will tell us whether those lakes are just chemistry, or whether they contain something more, something alive, something utterly unlike anything on Earth. The thought is both exciting and unsettling. Life that does not need water, that breathes hydrogen, that swims in methane, that might look at our warm water-covered Earth and think it seems impossible that anything could live there. The universe is stranger than we imagine, and Titan might be the proof. Mars was once a world of water. This is no longer speculation. It is established fact confirmed by decades of exploration. Rovers have found rounded pebbles that could only form in flowing water. Orbiters have mapped ancient river deltas where water once poured into lakes. Landers have analysed minerals that only form in the presence of liquid water. The evidence is everywhere. Mars had rivers, lakes, possibly even a northern ocean covering a third of the planet. The atmosphere was thick enough to keep water liquid. The temperature was warm enough for rain to fall. For at least 1 billion years, Mars was a wet world. Then something catastrophic happened. Mars lost its magnetic field. Without that protective shield, solar winds stripped away the atmosphere. The thicker air escaped into space. Pressure dropped, temperature plummeted. The water evaporated or froze. The rivers dried, the lakes turned to ice. The surface became the cold, dry desert we see today. This transformation took hundreds of millions of years, but once it was complete, Mars became hostile to surface life. Today the surface is bombarded by ultraviolet radiation. The thin atmosphere provides almost no protection. The soil contains perchlorates, chemicals toxic to most Earth organisms. Liquid water cannot exist on the surface. It would boil away in the low pressure or freeze solid in the cold. Any microbe exposed on the surface would be sterilised within hours. But life, if it emerged during Mars's wet period, did not necessarily die. It might have followed the water underground. On Earth, life exists kilometres below the surface. In gold mines in South Africa, researchers have found bacteria living 3 kilometres deep in solid rock. These organisms have been isolated from the surface for millions of years. They do not need sunlight. They do not need oxygen from the atmosphere. They get energy from radioactive decay in the rocks, from chemical reactions between water and minerals, from hydrogen released by geological processes. They live in complete darkness, reproduce slowly, survive in temperatures above 60 degrees Celsius. Their existence proves that life does not need a surface environment. It does not need photosynthesis. It just needs liquid water, energy, and the right chemistry. Mars has all of these things underground. Liquid water almost certainly exists in subsurface aquifers. We know this from radar measurements, from seasonal changes on the surface that suggest briny water seeping from underground, from modelling of Mars's geothermal heat. The planet is cold, but not completely frozen. Radioactive decay in the core generates heat. That heat rises. At certain depths, it is warm enough to keep water liquid, even with the freezing surface above. The chemistry underground is right for life. Martian soil contains the elements life needs: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. The minerals provide electron donors and acceptors for metabolism. Iron-rich rocks can support iron-reducing bacteria, like those found in Earth's subsurface. Sulfates can support sulfate-reducing bacteria. The energy sources are available: hydrogen from reactions between water and basalt rock, chemical gradients where different water types mix, even radioactive decay, providing a steady trickle of energy over geological timescales. If Martian life went underground billions of years ago, it could still be there, slowly metabolizing, reproducing on timescales of years or decades, surviving in refugia, isolated from the deadly surface. The question is how to find it. Our current rovers cannot drill deep enough. Perseverance can drill about 30 centimetres into rock, deep enough to get past surface sterilisation, deep enough to sample fresher rock, but nowhere near deep enough to reach subsurface water. The deepest drilling on Mars so far has been about 2 metres by the InSight lander's heat probe. That is not nearly enough. To find subsurface life, we need to drill tens or hundreds of metres. Future missions are being planned to do exactly this. Concepts exist for drill systems that can bore 100 metres down. for ice drills that can penetrate the polar ice caps and sample the water beneath, for rovers that can explore caves and lava tubes where underground water might be accessible. The most promising locations are not where we have been looking, not the equatorial regions where most landers have gone, but the mid-latitudes where ice has been detected just below the surface, or the poles where massive ice caps hide what might be liquid water at depth, or the slopes where recurring slope lineae appear. These are dark streaks that form on steep slopes during warm seasons. They fade during cold seasons. They appear year after year in the same locations. For years, scientists thought they were caused by liquid water seeping from underground. Briny water that stays liquid despite the cold because dissolved salts lower its freezing point. Recent analysis suggests they might not be water after all. They might be granular flows, dry sand sliding down slopes. But even if that is true, the presence of subsurface ice in those regions is confirmed. Where there is ice, there might be liquid water at depth. Where there is liquid water, there might be life. Mars also has caves. Dozens of them have been identified from orbit. Dark pits leading into underground voids, lava tubes where molten rock once flowed. Some of these caves might extend for kilometers underground. They would provide protection from radiation. Stable temperatures, possible access to subsurface water. If life exists on Mars, caves might be where we find it. But caves are dangerous for rovers. A wheeled vehicle cannot descend vertical shafts, cannot navigate rough terrain in darkness. Future missions might use rappelling systems, or flying drones that can explore the caves and send back images. The challenge is not just finding Martian life, it is proving that it is Martian life and not Earth contamination. Every spacecraft we send to Mars carries Earth microbes. Despite sterilization procedures, some bacteria survive. They are incredibly hardy. They can survive space radiation, extreme temperatures, years of dormancy. If we drill into Martian subsurface water and find bacteria, we need to be certain they are not Earth bacteria that contaminated our drilling equipment. This requires careful protocols, clean rooms, sterilization, documentation of every step, analysis that can distinguish Earth life from Mars life. The way to do this is through genetics. If Martian life uses DNA like Earth life, but with a different genetic code, different base pairs, different proteins, we can tell them apart. If Martian life does not use DNA at all, that makes it even easier. But if Martian life and Earth life share a common origin through panspermia, they might be nearly identical genetically. That would be scientifically fascinating, but would make it much harder to prove we found native Martian life rather than contamination. The Mars Sample Return mission will help answer these questions. Perseverance is currently collecting rock samples in Jezero crater, sealing them in tubes, leaving them for a future mission to retrieve. That retrieval mission will land on Mars in the early 2030s. Pick up the sample tubes, launch them into Mars orbit. A separate spacecraft will rendezvous with them, bring them back to Earth. The samples will arrive around 2033. They will be analysed in containment facilities, searched for signs of ancient life, fossils, organic molecules, patterns in the rock that could only be biological. These samples are from rocks that formed billions of years ago when Mars still had surface water. They might contain direct evidence of ancient Martian life, but they will not tell us if life exists on Mars today. For that, we need to go deeper, to drill into regions where liquid water exists now, to sample that water, to analyse it for living organisms. We need missions specifically designed to find current life, not ancient fossils, living, metabolizing, reproducing Martian organisms. These missions are coming. The technology is being developed, the drilling systems, the contamination protocols, the analysis instruments that can detect life in situ without bringing samples back to Earth. Within 20 years, we might have the answer, not from studying ancient rocks, from extracting water from Mars's subsurface, from analysing that water, from discovering whether Mars's underground biosphere is empty, or whether it is teeming with alien microbes that have been surviving in the dark for billions of years, waiting for us to find them. Enceladus should not be alive. It is tiny, only 500 kilometers in diameter, small enough to fit inside the borders of Texas. Its surface is frozen solid at -200 degrees Celsius. It orbits Saturn, nearly 1.5 billion kilometers from the Sun. By every measure, it should be a dead ball of ice, geologically inert, frozen solid throughout, unremarkable. But in 2005, the Cassini spacecraft discovered something that changed everything. Plumes of water vapor erupting from the South Pole, massive geysers shooting hundreds of kilometers into space, ice crystals, and water vapor streaming from cracks in the surface. This was not supposed to be possible. Enceladus is too small to retain heat from its formation, too far from the Sun to be warmed by solar radiation. Yet something was producing enough energy to melt ice and drive geysers more powerful than anything on Earth. The explanation is tidal heating. Saturn's gravity squeezes Enceladus as it orbits. Other moons pull on it too. These gravitational forces flex the interior. That flexing generates heat through friction. The same physics that keeps Europa's ocean liquid. But Enceladus is much smaller than Europa. The fact that it has active geysers suggests its interior is remarkably warm, possibly warmer than Europa's. Cassini flew through the plumes multiple times, analysing the material with onboard instruments. What it found was extraordinary. The plumes are not just water vapour, they contain salt, sodium chloride, potassium salts, carbonates. The same salts found in Earth's oceans. This confirmed that the water is coming from an ocean, salt water in contact with rock, not just melted ice, but a true ocean with dissolved minerals. Then Cassini detected organic molecules, not just simple carbon compounds, complex organics, molecules with chains of carbon atoms, aromatic hydrocarbons, nitrogen-bearing compounds. Some of these molecules are precursors to amino acids, the building blocks of proteins. The plumes also contained molecular hydrogen. This is crucial. Hydrogen is food for certain types of microbes. On Earth, bacteria called methanogens consume hydrogen and produce methane. They live at hydrothermal vents, in deep subsurface aquifers. Anywhere there is hydrogen and the right chemistry. They do not need sunlight. They do not need oxygen. Just hydrogen and carbon dioxide. If similar organisms exist on Enceladus, they could survive in the ocean, consuming hydrogen, producing methane, forming the base of an ecosystem. Cassini detected methane in the plumes as well. This could be produced by geological processes. Water reacting with rock at high temperatures can produce methane through serpentinization, but it could also be produced by life. The ratios and isotopes could tell us which. Future missions will analyse these in detail. The source of the hydrogen is significant. Models suggest it comes from hydrothermal reactions at the bottom of Enceladus's ocean. Hot water circulating through the rocky core, reacting with minerals, producing hydrogen and dissolving other compounds, then rising back to the ocean as hydrothermal fluids. This is exactly the process that supports life at Earth's deep ocean vents. If Enceladus has hydrothermal activity, it has energy for life. It has chemistry for life. It has everything life needs except one thing: time. Did Enceladus's ocean exist long enough for life to emerge? On Earth, life appeared relatively quickly once conditions were right, within a few 100 million years after the planet cooled enough for liquid water. But Earth is large. Its interior heat lasts billions of years. Enceladus is small. Does it have enough internal heat to maintain its ocean for billions of years? Or is this ocean a recent phenomenon? Models suggest the ocean might be ancient. Tidal heating is not a temporary process. As long as Enceladus orbits Saturn in its current configuration, tidal forces will generate heat. This could have been happening for billions of years, long enough for life to emerge, long enough for evolution to create complex ecosystems. The plumes make Enceladus uniquely accessible. We do not need to land on the surface. We do not need to drill through kilometers of ice. We can sample the ocean by flying through the plumes. A spacecraft could pass through them dozens of times, collecting particles, analysing them with advanced instruments, searching for direct signs of life. NASA is planning exactly this mission. The Enceladus Orbolander would orbit Saturn, make multiple flybys of Enceladus, fly through the plumes with instruments specifically designed to detect biosignatures, complex organic molecules arranged in patterns that suggest biology, amino acids with chirality consistent with life, fatty acids that could form cell membranes, perhaps even actual cells preserved in ice crystals. Then the spacecraft would land on the surface, deploy instruments to study the ice, analyze the chemistry, perhaps drill down to access fresher material erupted from below. The mission would not launch until the early 2030s at the earliest, but it is being developed. The science case is compelling. The technology is feasible. Enceladus is reachable. Some scientists argue we should send a mission sooner. That Enceladus is the best place in the solar system to search for life. Better than Europa because the plumes make sampling so much easier. Better than Mars because we know Enceladus has liquid water right now. Better than Titan because we understand water-based biology. If we want to find alien life in our lifetime, Enceladus might be our best chance. The question is what we would find. If Enceladus has life, would it be simple? Just bacteria metabolizing hydrogen? Or could it be more complex? Could evolution in Enceladus's ocean have produced multicellular organisms? Earth's oceans are full of complex life. Fish, octopuses, whales, all evolved over hundreds of millions of years. Enceladus's ocean might be older than Earth's oceans. It could have had more time for evolution. Not necessarily time measured in Earth years, but evolutionary time measured in generations. If organisms in Enceladus's ocean reproduce quickly, evolution runs quickly. A microbial generation might be days or weeks, millions of generations per million years, enough for significant evolution. We should not assume Enceladus life would be simple. This raises questions about ethics. If Enceladus has complex life, should we sample it? Should we disturb it? Should we risk contaminating it with Earth microbes? Most scientists believe the answer is yes. The scientific value is too great. The opportunity to study an independent origin of life is unprecedented, but we must do it carefully. Sterilization protocols, contamination control, minimizing our impact. The discovery of life on Enceladus would transform astrobiology. It would prove that life can emerge in subsurface oceans, that icy moons are habitable, that the outer solar system is not dead but alive. It would suggest that moons like Enceladus are common throughout the galaxy. Nearly every gas giant has moons. Many of those moons probably have subsurface oceans. If subsurface oceans can harbour life, then potentially millions of moons in our galaxy alone could have biospheres. The universe would be far more alive than we imagined, not just on planets in habitable zones around stars, but on moons everywhere, in orbit around gas giants, in orbit around ice giants, even around rogue planets drifting through interstellar space with no star at all. As long as tidal forces generate heat, as long as water remains liquid, life might find a way. Enceladus would tell us that life is not rare, not fragile, not restricted to Earth-like worlds in Earth-like conditions, but robust, adaptable, able to thrive in darkness, in cold, in isolation. The plumes of Enceladus might be more than geysers. They might be windows into an alien ocean, proof that we are not alone, evidence that life is written into the fabric of the cosmos, that wherever conditions allow, life emerges. We stand at the threshold of this discovery. The missions are being planned, the instruments are being designed. Within decades, a spacecraft will fly through those plumes with sensors capable of detecting life, and we will finally know whether Enceladus is just ice and water, or whether it is home to Earth's closest alien neighbours. Venus is hell. The surface temperature is 465 degrees Celsius, hot enough to melt lead. The atmospheric pressure is 92 times Earth's, equivalent to being a kilometre underwater. The air is mostly carbon dioxide, with clouds of sulfuric acid. Nothing we know of could survive there. Probes sent to the surface have lasted less than 2 hours before being crushed and cooked. Venus is the last place you would look for life, until 2020. In September 2020, scientists published a paper in Nature Astronomy. They had detected phosphine in Venus's atmosphere. Phosphine is a simple molecule, one phosphorus atom bonded to 3 hydrogen atoms. On Earth, it is produced 2 ways: by industrial processes, or by anaerobic bacteria in oxygen-free environments. Swamps, sewage, the intestines of animals. Everywhere phosphine appears on Earth, life is involved. There is no known natural geological process that produces significant amounts of phosphine. Not on Earth, not on Venus, not anywhere. The detection was made using telescopes on Earth and the James Webb Space Telescope. They analysed the spectrum of light passing through Venus's atmosphere. Phosphine absorbs specific wavelengths. Those wavelengths were missing. The signal was clear. Phosphine was present at concentrations of about 20 parts per billion. That might sound like a tiny amount, but it should not be there at all. Venus's atmosphere is harsh. Ultraviolet radiation from the sun should break phosphine apart within minutes. The clouds are sulfuric acid. They should destroy phosphine. Yet it persists. Something is producing it, constantly replacing what gets destroyed. The announcement was met with skepticism. Other scientists questioned the analysis, proposed alternative explanations, suggested the signal might be sulfur dioxide, not phosphine. The debate continues. New observations are planned, more sensitive instruments, better spectral resolution. We will know soon whether the detection was real, but the possibility has scientists reconsidering Venus entirely. Maybe life on Venus does not live on the surface. Maybe it lives in the clouds. This sounds absurd until you examine Venus's atmospheric layers. The surface is uninhabitable, but at an altitude of 50 to 60 kilometers, conditions are remarkably Earth-like. The temperature is around 30 degrees Celsius. Room temperature. The pressure is similar to Earth's surface, about 1 atmosphere. The clouds at this altitude contain water droplets. The composition is mostly sulfuric acid, which is hostile to Earth life. But some Earth microbes can tolerate acidic environments. Bacteria have been found in acid mine drainage, with pH values below 1. Archaea live in acidic hot springs. If microbes can adapt to acids on Earth, perhaps they could adapt to Venus's clouds. Carl Sagan proposed this possibility in 1967. He suggested that Venus's clouds might harbour floating microbes, organisms that never touch the ground, that reproduce in water, droplets, that get lifted by updrafts and fall with downdrafts in an endless cycle. On Earth, microbes live in clouds. They have been detected at high altitudes, reproducing, metabolizing, forming communities. If it happens on Earth, it could happen on Venus. But how would life get into Venus's clouds in the first place? Venus was not always hell. Billions of years ago, it might have been Earth-like. Models suggest Venus had liquid water on its surface for up to 2 billion years. Oceans, moderate temperatures, a thicker atmosphere that was not so hostile. Then a runaway greenhouse effect began. The sun gradually brightened. Solar radiation warmed Venus's surface. Water evaporated. Water vapor is a greenhouse gas. It trapped more heat. More water evaporated. The cycle fed on itself. Eventually, the oceans boiled away. The temperature soared. The atmosphere became the crushing, scorching hell we see today. But if life emerged during Venus's habitable period, it might have survived the transition. As the oceans evaporated, microbes could have been carried into the atmosphere. They could have adapted to living in water droplets in the clouds. As conditions worsened on the surface, the clouds became the only refuge. The organisms that survived were the ones that could tolerate acids, that could reproduce quickly in temporary droplets, that could form spores or protective structures to survive dry periods. Over billions of years, evolution shaped them into cloud dwellers, organisms whose entire life cycle occurs in the atmosphere. This is speculation, but it is plausible. Life is adaptable. It survives in extreme environments on Earth, in boiling water, in solid rock, in acid, in salt lakes so concentrated that water crystallizes. Venus's clouds are hostile but not impossible. The phosphine detection suggests something is happening. If not life, then what? Proposed alternatives include volcanic activity, releasing phosphine from the interior. But Venus shows little sign of active volcanism, lightning producing phosphine through electrical reactions. But the amounts do not match. Unknown atmospheric chemistry. But no model has successfully explained the observations. Life remains the simplest explanation. Not the only explanation, but the one that fits the data. Confirming this requires going to Venus, sending a probe into the clouds, collecting samples, analysing them for signs of life. NASA has approved 2 missions to Venus. Da Vinci will drop a probe through the atmosphere, measuring composition, searching for organic molecules, looking for isotope ratios that could indicate biology. Veritas will orbit Venus and map the surface, looking for signs of recent volcanic activity. Other missions are being proposed, balloons that float in the clouds for months, sampling the air, analysing droplets, searching for cells or complex organic molecules arranged in biological patterns. A life detection mission to Venus would be challenging. The sulfuric acid would corrode instruments. The temperature swings from freezing to hot as the probe descends. Sampling clouds while falling through the atmosphere requires clever engineering, but it is doable. The technology exists. The question is whether the science case is strong enough to fund it. The phosphine detection makes it stronger. If confirmed, it makes Venus one of the most interesting astrobiological targets in the solar system. Not despite its harsh conditions, but because of them. Finding life in Venus's clouds would expand our understanding of habitability. It would show that life can exist in atmospheres, that solid surfaces are not required, that life can survive environmental catastrophes and adapt to new niches. It would also raise questions about exoplanets. Many exoplanets orbit close to their stars. They are hot, possibly too hot for surface life. We have been ignoring them as candidates for life. But if Venus can have life in its clouds, these hot exoplanets might have cloud-based biospheres. We should be looking for biosignatures in their atmospheres, searching for phosphine or other gases that could indicate biology. The number of potentially habitable worlds would increase dramatically. Venus also teaches us about planetary evolution. Earth could have become like Venus. The same runaway greenhouse effect could have occurred here. The fact that it did not might be luck, or it might be biology. Life on Earth regulates the atmosphere. Plants consume carbon dioxide, produce oxygen. Algae in the oceans sequester carbon, bury it in sediments. Life keeps Earth's atmosphere stable. Venus lacked this regulation. Or perhaps Venus had life once, but lost it during the runaway greenhouse. The oceans boiled. Surface life died. Only the cloud organisms survived. A remnant biosphere clinging to existence in the only habitable zone left. If this is true, Venus is a warning. A preview of what Earth could become if we let greenhouse gases run unchecked. The discovery of life on Venus would be profound, not just for astrobiology, but for understanding our own planet, for appreciating how fragile habitability is, how quickly a world can transform from paradise to hell, and how life, once established, fights to survive even in the most hostile conditions imaginable. We will know soon. The missions are approved. The instruments are being built. Within a decade, a probe will descend into Venus's clouds, analyze the chemistry, search for life, and answer the question that has haunted us since the phosphine detection. Is Venus dead? Or is it inhabited by the solar system's most resilient organisms, floating in acid clouds, surviving against all odds, proof that life finds a way? Life on Earth exists almost everywhere we look. In deserts, in glaciers, in the deepest ocean trenches, in boiling hot springs, in solid rock kilometers underground. Every time we explore a new extreme environment, we find life. This has changed how we think about habitability. The limits keep expanding. The definition of habitable keeps widening. And it suggests we have been thinking too narrowly about where to look for life in our solar system. Consider the deep subsurface. On Earth, microbes live in rock formations several kilometers below the surface, in aquifers isolated from the surface for millions of years, in cracks and pores in solid basalt, in coal seams, in oil reservoirs. These organisms do not need sunlight. They do not need atmospheric oxygen. They get energy from chemical reactions, from hydrogen released by interactions between water and rock, from radioactive decay, from electron gradients between different minerals. Some of these organisms reproduce incredibly slowly. Cell division might take years or decades. Metabolism operates at a crawl, but they survive. They maintain their biochemistry. They repair damage. They constitute what scientists call the deep biosphere, and it is massive. Estimates suggest the deep biosphere contains as much biomass as all life in the oceans, possibly more. Most of Earth's life by mass might exist underground, invisible, inaccessible, unknown until we drill deep enough to find it. This has profound implications for Mars. If Earth's deep biosphere is so extensive, Mars could have an equally extensive subsurface ecosystem. Martian life does not need to be on the surface. It does not need to be anywhere we can easily reach. It could exist kilometers down, in aquifers warmed by geothermal heat, in pockets of liquid water between frozen layers, in rock formations where chemistry provides energy. Finding it would require drilling deeper than we have drilled, exploring regions we have never explored, going beyond the easy targets. The same logic applies to other worlds. Enceladus and Europa have subsurface oceans, but they also have rock cores. Water circulates through those cores, interacts with minerals, creates chemical gradients. Could life exist not in the open ocean, but in the rocks beneath, in porous minerals where water flows, in cracks where different chemistries meet? On Earth, some of the most interesting subsurface life exists at interfaces, where oxygenated water from the surface meets reduced water from below, where different aquifers mix, where minerals dissolve and precipitate. These interfaces create energy gradients. They drive metabolism. They support communities. Icy moons could have similar interfaces, boundaries between different ice phases, regions where the ocean meets the rock, cracks in the ice where fresh water from above meets salty water from below. These could be hotspots for life, more productive than the open ocean, more diverse, more interesting. Then there are truly exotic possibilities, life that does not use water at all. On Titan, we have talked about methane-based life. But there are other possibilities. Ammonia-water mixtures stay liquid at much lower temperatures than pure water. Some of Saturn's and Uranus's moons might have ammonia-water oceans beneath their surfaces. Life using ammonia water as a solvent would be different from Earth life. Different chemistry, different proteins, but potentially viable. Hydrogen fluoride is another possible solvent. It stays liquid at very low temperatures. It can dissolve many compounds. Life using hydrogen fluoride would be radically different, perhaps impossible, but we should not assume water is the only option. Formamide is another candidate. It stays liquid across a wide temperature range. It can participate in complex chemistry. Some scientists have proposed that life on Earth might have used formamide before transitioning to water. If formamide-based life is possible, it could exist on worlds too cold or too hot for water-based life. Then there are non-chemical possibilities. Could life be based on something other than chemistry? Plasma life has been proposed. Organized structures in ionized gas. They could form in the atmospheres of gas giants, in stellar coronas, in nebulae. They would not be biological in any sense we recognize, but they might exhibit complexity, growth, reproduction, evolution, patterns that emerge from physical processes rather than biochemistry. This is highly speculative. Most scientists doubt plasma life is possible, but we should keep an open mind. Silicon-based life is often proposed in science fiction. Silicon is similar to carbon. It forms 4 bonds. It can create chains and complex molecules. But silicon chemistry is less versatile than carbon chemistry. Silicon bonds are weaker at high temperatures, stronger at low temperatures. Silicon-based life might exist on very cold worlds, using silicon polymers instead of carbon chains. This would be so alien we might not recognize it. Another exotic possibility is life in subsurface oceans of water at very high pressures. On large icy moons or ocean worlds, the pressure at depth might be so great that water takes on exotic properties. High-pressure ice forms. Ice 7, Ice 10. These phases are solid but have unusual structures. Could life exist at the boundary between liquid water and high-pressure ice, using the unique chemistry that occurs there? We do not know, but we should look. The search for exotic life requires different strategies. We cannot just look for DNA or proteins. We need to look for complexity, organization, patterns that could not occur naturally, disequilibrium chemistry, ratios of elements or isotopes that suggest biological processing. This is challenging. It requires broad thinking, open minds, willingness to consider possibilities that seem unlikely, but the payoff would be immense. Discovering life that does not use DNA, that does not use proteins, that has a completely independent origin and operates on completely different principles. That would tell us life is not one solution to the problem of metabolism and reproduction. It is many solutions, each adapted to local conditions, each arising independently wherever conditions allow. The universe would be full of life, But that life would be more diverse than we imagined, more strange, more wonderful. Our solar system is the testbed, the place where we can explore these possibilities, where we can send missions to exotic environments and see what we find. In the next few decades, we will sample subsurface oceans, analyze cloud droplets, drill into ice and rock, explore lava tubes and caves. Each mission will either find life or constrain the possibilities. Each discovery or non-discovery teaches us something. Gradually, we will map the boundaries of habitability, the limits of where life can exist, and perhaps discover that those limits are much wider than we thought, that life is everywhere, hiding in plain sight, waiting for us to look in the right places with the right tools. The next 2 decades will answer the question. We have the technology, we have the missions, we have the instruments. Within 20 years, we will know whether alien life exists in our solar system. Multiple missions are currently active or planned. Perseverance is collecting samples on Mars. Europa Clipper launches in 2024. Dragonfly launches in 2027. Da Vinci will probe Venus's atmosphere. Future missions will drill into Mars's subsurface, sample Enceladus's plumes, explore Titan's lakes and seas. Each mission is a roll of the dice, a chance to find life. The odds are uncertain, but not zero. Some scientists are optimistic. They point to the abundance of habitable environments, to the resilience of life on Earth, to the speed at which life emerged here. If life arose on Earth within a few 100 million years, it should arise anywhere conditions are suitable. Europa has had a liquid ocean for billions of years. Enceladus has hydrothermal vents. Mars had surface water for 1 billion years. Titan has complex organic chemistry. The ingredients are there. The environments exist. Life should emerge. Others are pessimistic. They argue that abiogenesis, the origin of life, might be extraordinarily unlikely. That Earth might be a statistical fluke, that the jump from chemistry to biology requires so many unlikely steps that it almost never happens. If this is true, we could search the solar system for centuries and find nothing. Not because life cannot exist there, but because it never started. The debate will be settled by observation, not by argument. We need to look and see what is there. The first detection might be indirect. Chemical signatures in Europa's plumes. Isotope ratios inconsistent with geology. Patterns in Titan's lakes that suggest biology. Phosphine in Venus's clouds confirmed and explained. These would not be proof, they would be hints. Tantalizing suggestions that require follow-up. The gold standard would be direct detection. Finding cells, seeing structures under a microscope that could only be biological. Analyzing complex organic molecules arranged in patterns that evolution produces. Detecting metabolism. Observing reproduction. This requires sophisticated instruments, microscopes that can work in extreme environments, spectrometers that can identify thousands of compounds, DNA sequencers that work on non-DNA life, chemical analyzers that can detect patterns rather than just individual molecules. These instruments exist. They are being miniaturized, being hardened for space, being tested in Earth's most extreme environments. Within a decade, we will deploy them on other worlds. The strategy is to start with the most accessible targets. Enceladus's plumes, Europa's potential plumes, Mars's near-surface ice. These locations can be sampled with flyby missions or simple landers. If we find nothing, we go deeper. Drill into Europa's ice. Explore Mars's subsurface. Send submarines into Enceladus's ocean. Deploy balloons into Venus's clouds. Gradually expand our search until we either find life or exhaust the possibilities. Some environments will require decades to explore properly. Titan's lakes need submarines or boats, sampling multiple locations, analysing sediments, looking for organisms or their remains. The deep subsurface of Mars needs drilling rigs, capability to go hundreds of meters down. Sample aquifers, analyse the water and rock. These missions are expensive, technically challenging, but they are being planned. The technology is being developed. The science case is strong. If there is life in our solar system, we will find it. The question is, what happens when we do? The discovery would be announced carefully, verified by multiple teams, confirmed beyond doubt. The sample would be analysed exhaustively, every possible alternative explanation eliminated. Only after all that would NASA or ESA make a public announcement. The reaction would be enormous. Immediate, global, proof that we are not alone, that life exists beyond Earth, that the universe is inhabited. The scientific implications would be profound. If we find a second genesis, a completely independent origin of life, it tells us abiogenesis is not impossibly rare, that wherever conditions allow, life emerges, that the universe must be full of life. If we find that life is related to Earth life, sharing a common origin through panspermia, it tells us life spreads between worlds, that biospheres are not isolated, that life, once started, propagates across space. Either answer transforms our understanding. But there are also philosophical and cultural implications. Religions would need to incorporate alien life into their worldviews. Some would find this easy, others would struggle. Humans would need to confront our cosmic insignificance. We are not the only life, not even in our own solar system. Just one branch on a tree that includes Martian microbes or Europan swimmers. Ethics would become complicated. Do we have the right to disturb alien ecosystems, to sample them, to potentially contaminate them? Most scientists believe the answer is yes. The knowledge gained outweighs the risks, but not everyone would agree. There would be debates, protests, calls to leave alien life undisturbed. Then there are the practical considerations. If we find life on Mars, human colonization becomes more complicated. We cannot just terraform Mars if it has native life. We would be destroying an alien biosphere, building our civilization on top of theirs. Some would argue this is unacceptable, that Mars should be protected. Others would argue humans matter more than Martian microbes, that we have the right to expand, that life on Mars would benefit from being studied and preserved by us. These debates would be fierce. They would shape space policy for generations. But they would be good debates to have, because they would mean we found something, proof that we are not alone. The alternative is silence. Decades of searching, missions returning empty-handed, samples showing no signs of life, chemical analyses finding only geology. This would be disappointing, but also informative. It would tell us abiogenesis is rare, that Earth is special, that we might be alone in the solar system, even in the galaxy. This would make us precious, the only known life, responsible for preserving and spreading life in a sterile cosmos. It would change how we see ourselves, make us appreciate Earth more, recognize how unique our biosphere is. Some would see this as lonely, others as empowering. We are the universe's way of knowing itself, the only consciousness, the only witnesses, the only hope for life to spread beyond one world. We do not know which answer we will get, whether our solar system is inhabited or sterile, but we will know soon. The missions are launching, the instruments are ready, the search is underway. In 2030, Europa Clipper will arrive at Jupiter, begin its flybys, sample the plumes. In 2034, Dragonfly will land on Titan, begin exploring. In the early 2030s, Mars samples will return to Earth, be analysed. Later in the 2030s, new missions will drill into Mars, sample Enceladus, explore Venus. By 2040, we will have searched the major targets, sampled the most promising environments. We will either have found life or significantly constrained where it could be hiding. By 2050, we will know. The answer to one of humanity's oldest questions, are we alone? The search is not happening on distant exoplanets. It is happening here, in our cosmic neighbourhood, on worlds we can see with backyard telescopes, on moons that have been familiar since Galileo first pointed his telescope at Jupiter. The closest aliens might be closer than we think, hiding beneath ice, floating in clouds, swimming in subsurface oceans, surviving in Martian aquifers, waiting for us to find them. The universe is vast, but the search begins at home, in our own solar system, where alien life might be hiding in plain sight, and we are finally learning where to look.